So for our study, in order to determine mechanisms of acquired resistance to KRAS G12C inhibitors, we included patients that were treated on adagrasib or MRTX849 monotherapy clinical trials. This included 23 patients with non-small cell lung cancer and 7 patients with colorectal cancer. And for patients included in our analysis, we took a look at either repeat biopsies or circulating tumor DNA obtained at the time of disease progression on adagrasib and compared that to samples taken prior to adagrasib therapy initiation in the hopes of trying to identify genomic or histologic changes that developed over the course of treatment to determine the mechanisms of resistance to the drug. So in our study, we identified several novel mechanisms of resistance to adagrasib. We found several mutations in what's called the switch II binding pocket of KRAS. So we found novel alterations at KRAS amino acid position 95, so histidine 95 mutations. We found tyrosine 96 mutations, arginine 68 mutations, as well as novel mutations affecting other portions of KRAS. So we found that the KRAS G12C amino acid, that's the cysteine residue that drugs like adagrasib and sotorasib covalently bind to, that we found new mutations at KRAS G12, also at codon 13, and other KRAS alterations. So we found a number of acquired mutations within KRAS. We call those on-target mutations, which also included acquired amplification of the G12C allele. And then we found several off-target mechanisms of resistance, including mutations in genes like BRAF or NRAS or MEK, also called MAP2K1, as well as a number of acquired gene fusions in genes such as BRAF, RET, RAF1, ALK, and others. So by studying these samples, we found many mechanisms of resistance. In some patients, we found a single mechanism of resistance, and in others, we found multiple concurrent or simultaneous disease resistance mutations. We also identified a couple of cases that had histologic transformation from lung adenocarcinoma to lung squamous cell carcinoma. And in those cases, we did not find any acquired genomic alterations to explain the mechanism of resistance to the drug. So we think these findings have several implications for drug development. I think this is the first characterization of how cancers can develop resistance to this class of drugs clinically and in patients. And so we hope that this will inform subsequent trial design to either develop novel inhibitors to hopefully overcome some of these resistance mechanisms, or to develop rational combination strategies that can be used up front in combination with KRAS G12C inhibitors to hopefully delay the emergence of resistance, or once resistance emerges, to use those combinations ideally to overcome resistance. So our hope is that these findings will really inform subsequent clinical trial design and drug discovery. I just wanted to thank, this was a real collaborative effort involving many different institutions as well as industry partners. And I wanted to especially thank my close collaborator, Dr. Andrew Aguirre at the Dana-Farber Cancer Institute. In his lab, they did a very exhaustive mutagenesis screen of KRAS to try to identify additional mutations that could confer resistance to drugs like adagrasib and sotorasib. And so we also included a lot of preclinical data from his laboratory analysis, and I really wanted to acknowledge his seminal contributions to this study.